So good morning, everybody, and thanks for coming along to today's talk on the weird and wonderful world of microbiology. So my name is Shane Dillon. I'm a microbiologist here at Trinity, and I'm here to give you an overview of the broad topic of microbiology and hopefully persuade you to come to Trinity to study science and maybe specialize in microbiology. So I work in this building. You might have seen it on your way in today, beside the cricket pitches here. So this is the microbiology department, which was built in 1953, and it's recognized as being one of the best microbiology departments in not only in Europe, but perhaps in the world. A number of key microbiology discoveries have actually been made in this department, and it was built by funding from the Guinness family. So it's got a very good reputation in a short space of time. In the last 70 years, as I said, a number of key microbiology discoveries have been made in this department. And this is where a number of internationally successful research groups conduct microbiology research on a number of topics today. Okay, so what is microbiology? You might be interested to for me to give you a broad description of what actually microbiology is. So we typically study organisms that are too small to be seen with the naked eye. So this encompasses a range of organisms, from bacteria, like E. coli imaged here, to viruses, to fungi, such as yeast, algae. So it's a picture of algae here, to protozoa. For example, the protozoa that causes malaria is studied in our department. We study viruses in our department, like the virus that causes the influenza virus. And we also study fungi. So this is an image of fungi here. So in particular, yeast that are involved in brewing. So we study a range of microorganisms that are too small to be seen with the naked eye in our department. I just want to take you on a brief history of the field of microbiology. And it probably began in the late 1600s with this Dutch individual, Anton van Leeuwenhoek. So he developed the first microscope, and he actually was able to visualize bacteria, for example, in stool samples and in water samples, and he essentially discovered bacteria. But this was all due to serendipity. He was actually a linen trader, and he wanted to judge the quality of the cloth he was buying. So he developed this magnifying lens, which allowed him to count the number of threads in the linen and other cloths that he was buying. But as I said, by developing this magnifying lens or microscope, he was able to discover bacteria. Up until then, nobody had ever seen bacteria. So he's considered the first microbiologist. So we know that bacteria, as I said, are very small, but this maybe will put things into context. Where the word microbiology comes from is that bacteria obviously are very small. This is a magnified view of a tip of a pin, which is about 20 microns in diameter across the tip of the pin. So we all know that a pin is incredibly small, but you can see when you magnify this pin that the bacteria are even smaller. So this is bacteria on the tip of a pin, and you can see that they're approximately 2 microns in size. So obviously bacteria are very small. And the organism I work on is Salmonella typhimurium, which causes food poisoning. So it's maybe, like these bacteria here, it's about 2 microns in length. As I said, you can't see it with the naked eye. So these microorganisms are not visible to the naked eye. As I said, we require microscopic methods to visualize these microorganisms. So why are microorganisms important? So pretty much everything on planet Earth depends on microbes. We know that they make up about 60% of the Earth's biomass. They outnumber all species and as I said, make up most of the living matter on Earth. Without microbes, we wouldn't be here. They play key roles in the cycling of essential elements like carbon and nitrogen, which allow us to inhabit our planet. They also generate half of the oxygen we breathe as well, along with plants. Microbes generate a huge amount of the oxygen we breathe. So without them, I wouldn't be here. We certainly wouldn't be listening to this talk without the vast number of microbes on planet Earth. So we know that they can live everywhere, and like us, they can be found in a wide range of habitats. So we can isolate microbes 1.5 kilometers underneath the ground where there's no sunlight, very little in terms of nutrients. We can actually isolate bacteria or microbes from the sky. So scientists have discovered microbes living in clouds, on ice, in hydrothermal vents, these extremely warm hydrothermal vents, and many kilometers down within the sea, we can isolate different types of microbes. And we know that these microbes help us in so many ways. I'll just take you through a few examples of what microbes do for us in our daily lives. So we know that the pictures of our dairy cows here. So we know that without microbes, dairy cows wouldn't be able to use grass as an energy source. So we know that there are vast numbers of microbes in the rumen of the cow, which break down the cellulose in grass. And the cows don't have the enzyme to break down this cellulose, so they require bacteria in their rumens to produce enzymes to break down cellulose. So without microbes, these ruminants wouldn't be able to break down grass, and obviously we wouldn't be able to derive any of the products we do from cows. Amazingly, microbes can be used to degrade explosives as well. There's examples of in places where ammunition is kept, that TNT can leak out of ammunition and is a serious problem for contaminating the soil. We know there's a species of bacteria called Clostridium bifurmentans, which can actually break down TNT. So it can use TNT essentially as an energy source and is able to metabolize TNT and clean up these areas where you have contamination of the soil with TNT. We know when you're sitting down over the Christmas period to have some chocolates, just bear in mind that without bacteria, you wouldn't be able to eat those chocolates. So we know that chocolate comes from the seeds of the cacao tree, and they produce these pods which contain the seeds, and actually the seeds are retrieved by fermenting these pods with a combination of microbes, so yeasts, lactobacilli and acetobacter play a key role in breaking open these pods, and in particular a type of bacteria called lactobacillus secretes an acid to break apart these pods, so we can access the seeds and make chocolate. There's also some interesting stories in terms of reviving ancient bacteria. So there's a Sleeping Beauty story for bacteria as well. In 1995, a number of scientists revived these bacteria that were believed to be between 25 to 40 million years old. So they were preserved in the stomach of a bee, and the bee was preserved in tree sap. And it's believed that this idea was used by Steven Spielberg for the Jurassic Park idea, where he created dinosaurs after the DNA was extracted from mosquitoes trapped in tree sap in the same way, and was preserved for millions of years. We also know that bacteria can act as natural insecticides. Again, there's a species of bacteria called Bacillus thuringiensis, which produces specific proteins that are toxic to insects but aren't toxic to humans. And scientists have cleverly transferred these specific bacillus genes into crops, so they produce natural toxins which are fatal to these insects, but obviously don't cause us any harm. So there are a huge number of uses that microbes can do in our everyday lives. Another use is we know that a species of the soil bacterium Pseudomonas is able to use caffeine as an energy source as well, so it can metabolize caffeine. And it's been suggested that this bacterium could detoxify the toxic material that's left after we separate coffee beans from the coffee berries in the manufacturing of coffee beans. So there's a serious issue with water pollution during the process of separating the coffee beans from coffee berries. And again, bacteria may come to the rescue by being able to metabolize these waste products as well. So I just want to show you some examples in a short YouTube clip of the huge amount of natural diversity around us. So this, I'm going to show you an image or play you a clip of some dark field microscopy of a natural environment. So in this clip, you're going to see some of the amazing number of microbes that are just present in simple pond and lake water samples. So you'll see in this clip a number of crustaceans, mosquito larvae, and other microbes that are present all around us just simply in pond and lake water. So I'll play this clip for you now. Okay, so I hope that just illustrated some of the amazing microbial diversity just simply in a sample of pond water. So it's these amazing microbes are all around us, as I said. So it's a very natural, a very interesting example of microbes in the natural environment. So as I said, back to us maybe in terms of how bacteria impact upon us. We know that bacteria live all over, so I'm just showing you some images of some agar plates where we're able to culture different types of bacteria. If we take samples, for example, from earwax, from sweaty socks and nasal secretions, you can see the diversity of microbial organisms that live all over us as well, and they have a huge impact on us. But the vast majority of microbes are what we would describe as friendly bacteria, and a huge number of bacteria colonize the human gut and actually protect us against a variety of diseases and provide a vast number of beneficial effects. So we know that friendly E. coli produce a number of vitamins. They produce vitamin B7 and vitamin K in your gut that we can't produce without these friendly bacteria. And we would be in serious problem. As I said, we don't have the enzymes required to produce these important vitamins. But we also know that they break down indigestible carbohydrates, for example, in cellulose in our diet. Again, we don't have the enzyme that's required to break down indigestible carbohydrates, but bacteria do that job for us in return for access to nutrients as well within the human gut. So we've all seen the examples of pots of friendly bacteria, the Actimel drinks, the probiotic drinks that are typically have lactobacillus in these friendly bacteria drinks. And scientists and microbiologists in particular are working to understand how these friendly bacteria give us important health benefits. But I just wanted to give you maybe an outline of how many friendly bacteria are actually inside you, give you an idea of exactly how many bacteria you have inside the human body. So there are about 1,000 trillion bacteria inside the human body. That's 1,000 million bacteria. And in fact, it's probably, if you weighed all those bacteria, they'd weigh as much as a can of baked beans. So approximately a kilogram of bacteria inside the human body. And they probably outnumber us 1,000-fold. There's maybe 1,000-fold more bacterial cells than human cells inside you. So now I'd like to briefly talk to you about some of the pathogens and most of the research in the microbiology department is on trying to understand bacterial pathogens. We know that these bacterial pathogens come in a range of shapes, in interesting shapes. So we have spherical bacteria, so Streptococcus pneumonia, which causes pneumonia. We have rod-shaped bacteria that I work on, like E. coli and Salmonella, which can cause food poisoning. We have spiral-shaped bacteria like Campylobacter jejuni, which causes food poisoning as well. And we also have comma-shaped bacteria like Vibrio cholera, which is the causative agent of cholera. So we know that, as I said, there's a wide range of shapes and morphology in the bacterial pathogens. With all these bacterial pathogens, it's pretty amazing that you're not dead yet. And you may know that actually the life expectancy has doubled in the past 150 years. But the question you might ask yourselves is why has our life expectancy doubled in the last 150 years? So if you went back to 1848, you could be expected, your average life expectancy was approximately 40 years of age. Today in Ireland, the average life expectancy is about 80 years of age. And if I just illustrate to you why this may be, so if we break down the average life expectancies between 1837 to 1841, you see that the highest life expectancy was for a person who was of the gentry class working in agriculture. Their average life expectancy was about 50. Whereas, for example, a labourer or a servant working in the commercial industry could expect to live to the age of 15 during this period. So individuals working in manufacturing and commercial were typically associated with the cities, and there was vast numbers of people living in the cities in very unhygienic conditions, whereas individuals working in agriculture out in the countryside tended to have a higher life expectancy than individuals living in cramped and overcrowded cities in unhygienic conditions. So what was killing everybody at the time? Well, it was, as you can expect, it was a number of microbes. So the leading causes of death around 1900, 4 of these were caused by bacteria. So diphtheria, diarrheal disease, tuberculosis, and pneumonia, indicated here in red, were among the leading causes of death around 1900 in the US. And if we fast forward to 1998, nearly 100 years later, we see that maybe pneumonia is the only one that makes this list of leading causes of death in developed countries. And they've been replaced by, for example, cancer and heart disease as the leading causes of death in developed countries. And the question is why? Why is that so? So if we go back to, as I said, to pre-1850, people believed that disease was transmitted by miasma, so a poisonous vapor or mist was believed to transmit a lot of diseases. This image shows you just what people believed at the time, that cholera was transmitted on the wind. Obviously now we know that it's caused by a type of bacteria, but back then, as I said, they didn't know that bacteria caused these diseases. And if you look at a typical public health notice from the time on how to prevent cholera in New York in 1832, it typically suggested that people avoided getting wet, kept warm, don't sleep in a draft, and avoided alcohol to prevent them from getting cholera. It wasn't until the founder of modern public health came along, a microbiologist called Robert Koch, who discovered in the late 1800s the causal agents of most of these diseases that were killing people. So he discovered that tuberculosis was caused by a specific bacterium called Mycobacterium tuberculosis. He discovered that cholera was caused by Vibrio cholera, and that anthrax was caused by Bacillus anthracis. So he's the founding father of public health, and he made a lot of these discoveries in the late 1800s and really, really helped to eradicate a number of these leading killers. So as I said, these diseases no longer threaten humanity because microbiologists have actually discovered the causal agents. We know that smallpox is caused by a specific virus. We know that typhoid is caused by a specific bacterium, Salmonella typhi. And we also know that diarrhea is caused by E. coli. These were big killers before we discovered the causal agents and ways to treat these infections. So I said these diseases no longer threaten humanity because microbiologists have actually discovered the causal agents and how to combat them. So we've developed a vaccine against smallpox. We know how to vaccinate against typhoid fever, and sanitary conditions prevent a lot of these cases of typhoid fever. And we know again for diarrhea that sanitation and food safety are important for preventing diarrhea. So these were major killers before microbiologists discovered how to combat these infections. So if we look at the deaths from infectious diseases in the US from 1900 to 1996, we've seen that there's been a dramatic decrease in deaths from infectious diseases. Again, this is due to research from microbiologists that suggested that we should chlorinate the water supplies to improve the sanitation of the water supplies. Although you'll notice here there was a spike due to the Spanish flu in 1918, which killed millions of people worldwide. But the use of penicillin in the 1940s, again, saw that there was a continued decrease in the number of deaths from infectious diseases in the past century. And again, scientists, microbiologists introduced vaccines in the late 1960s, again helped eradicate a lot of these leading killers. Of course, hygiene is very important. Nowadays we know that's the best prevention against most of these diseases. Obviously, to scrub these pathogens off your hands is the easiest way to prevent a lot of these diseases. So I just want to briefly take you through more recent data that suggests that E. coli strains, in particular outbreaks of E. coli strains, suggest that microbiologists haven't solved all of these problems. We know that in 2011 there was an outbreak of E. coli, very dangerous E. coli, in beansprouts that was able to kill a number of people, killed several hundred people in Germany. You probably didn't suspect that beansprouts could kill you, but as I'll briefly describe to you, in the summer of 2011, we know that there was an outbreak of E. coli between the months of May and June in a number of regions around Germany. And this was due to the emergence of a new hybrid E. coli strain. So as I said, bacteria can transfer genes between themselves. So there was a creation of a hybrid E. coli strain. So normally we can deal with two types of E. coli strains. So we have an enteroaggregative E. coli strain, and these two strains usually cause specific diseases. But in the summer of 2011, you had a hybrid strain formed between these two different strains. As I said, these strains both have individual tools that allow them to cause specific illnesses. But as I said, the E. coli, German E. coli outbreak was due to the creation of a hybrid strain where you had the transfer of a toxin from this E-hex strain into this enteroaggregative E. coli strain, and it created this hybrid 0104 strain in 2011, which, as I said, which killed several hundred people in Germany. So what happened with this hybrid strain? So as I said, normally you have this enteroaggregative E. coli, which is well adapted to adhering to the human gut wall, and you have this separate E-hex strain, which, as I said, causes a separate illness, and it produces a Shiga toxin. So you had a hybridization event where you had the transfer of the Shiga toxin into this enteroaggregative E. coli strain that produced the hybrid and very dangerous strain in 2011. So this had two key properties. It was adherent, so it could adhere to the human gut wall, but it was also toxic because it produced this Shiga toxin. So it led to nearly, over 840 cases of kidney damage, severe kidney damage due to the presence of this toxin. As I said, there were over 3,000 cases were diagnosed in a short space in Germany, and it led to 50 deaths at least in individuals. So this was quite a serious hybrid strain. So again, microbiologists haven't solved all of these problems because we continue to see the emergence of these new hybrid strains all the time. So where did the E. coli come from? The blame game started. Spanish cucumbers were blamed initially, but they weren't found to be the source of the E. coli outbreak. Then German lettuce was blamed and was destroyed, but that wasn't the source of the outbreak. And finally, it was tracked down to German beansprouts, but the seeds were actually came from Egypt and were produced under very unsanitary conditions. So again, microbiologists were able to come to the rescue and identify the cause and how to combat this outbreak. As I said to you, bacteria have a huge impact on humans. And you've probably heard of the antibiotic penicillin, and that was discovered in 1928 by the scientist Alexander Fleming. It was discovered by chance. He left out some plates of bacteria and saw that a mold grew on these plates, which produced this penicillin antibiotic. And that was first used in World War II to treat patients. So thanks to the development of these antibiotics, in the last 70 years, we were able to treat most of these bacterial infections. We've seen in the last few years that there's actually been an increase in bacteria that are resistant to these antibiotics due to overuse of antibiotics. Drug resistance is on the rise. As I said, this hybrid strain that I talked to you about is resistant to most antibiotics as well. As I said, we've been using antibiotics in huge amounts during the past 70 years, and that's led to the selection of strains of bacteria that are resistant to most antibiotics. You've probably heard about the superbugs, the MRSA and the C. diff that are resistant to antibiotics in hospitals and present a huge challenge to people treating bacterial infections. So we need to develop new antibiotics, and that's the challenge for the next generation of microbiologists, is to meet the challenge of these new pathogens that are emerging all the time, to understand how new viruses and bacteria cause disease. And I haven't talked to you about the Ebola virus, but obviously that's been in the news quite a lot recently. And again, microbiologists are on the cusp of developing a vaccine to the Ebola virus, which is being tested at the moment. So we do need the next generation of microbiologists to, as I said, to understand how bacteria and viruses cause disease. So I'd suggest if you're interested and have natural curiosity in your environments, that you do actually consider coming to Trinity to study science. And in your third and fourth years, you can specialize in the field of microbiology. It presents a diverse number of career options. Again, if you come down and talk to individuals at the microbiology stand, we can advise you on what opportunities are open to microbiologists. The graduates from the department have gone on to pursue careers in a wide variety of areas, from continuing in research and like I have to working in industry and to working in other areas of science as well. So please come along to the microbiology stand, and please talk with myself and other members of the department at the stand. So thank you for listening.